And welcome again to Chapter 12. This is the addendum to the second lecture in Chapter 12 on muscles. The two big topics we're going to be dealing with today are excitation-contraction coupling and smooth muscle contraction. So we're going to use smooth muscle to differentiate from skeletal muscle. So that'll be later on in the lecture. Just a few things to remind you about part one of muscle physiology is that we're always trying to sustain ATP levels. We only have a couple of seconds of ATP at any given time in our muscle fibers. So we're always banking energy where possible. So for instance, when we're not contracting our muscles, what we're going to do is we're going to shift that phosphate onto creatine and create phosphocreatine. So if ATP levels begin to drop, what will happen is we'll shift using substrate level phosphorylation ATP or the phosphate off of phosphocreatine to create more ATP. And why is that so important? Because of these three major reasons. We can't afford to lose ATP or the cell will begin to die. So certainly contraction is an important role for ATP. We hydrolyze ATP in order to cock the myosin head. We also need ATP to relax. So it's kind of an interesting dichotomy. So you say, hey, we need muscles to contract, we need ATP to relax. Why do we need ATP to relax? Is because we're pumping calcium out of the cytosol. We're going to look at that today when we look at excitation-contraction coupling. We actively remove calcium from the cytosol in order to achieve relaxation. And thirdly, the cell does more than contract. It needs nutrients, it needs to do other things, maybe need to signal a little bit, and so we have to sustain membrane potential. If we don't have membrane potential, we don't have osmotic regulation, for instance, the cell will begin to swell and die. So we have very, very precise needs for ATP inside the cell. So think about these three major reasons here that I've highlighted, why we would keep ATP at very, very tightly controlled levels. A little bit more about fatigue. We did talk about this earlier. If we have extended submaximal exercise, like for instance, long distance walking or running or continuous activity throughout the day, what we're going to see, one of the primary drivers is going to be depleting glycogen. If we have less glycogen, we have to obtain glucose or other sugars from the blood, or we have to rely on fat stores or other proteins in order to achieve oxidative phosphorylation. If we have short duration maximal exertion, like for instance doing a pull-up, a body weight push-up, the main driver here is phosphate release as well as calcium issues. Either we have decreased calcium release because we already have calcium released out, or we have calcium leak. That is to say, we lose calcium to the extracellular fluid, and so we just don't have enough calcium in order to achieve continuing contraction. So those are kind of the two major mechanisms in maximal exertion. Near maximal exercise, that is to say sustained or otherwise near maximal exercise, what we often see is issues with potassium, which may lead to things like changes in sodium potassium ATPase activity. And so these are, you can see different scenarios of muscular contraction over time that lead to these mechanisms of fatigue. And you'll notice that we don't have lactic acid in here. Moving on, we're going to now talk about how muscle fibers utilize energy, and we tend to see what we call a fast twitch mechanism and a slow twitch mechanism. So there are two major divisions, fast and slow twitch. In the fast twitch division, we have two flavors of fast twitch. We have sort of a medium, shall we say, it's a fast twitch oxidative glycolytic, which means that it can shift from oxidative or glycolytic. We have a true glycolytic, which is using basically substrate level phosphorylation to achieve rapid kinetics. And then we have our slow twitch, which relies primarily on oxidative phosphorylation. Slow twitch take longer to achieve maximal force. They take a few more minutes. That's because there's a sort of a delay as we ramp up oxidative phosphorylation. Fast twitch fibers are different from slow twitch in that they can develop tension faster because their myosin can hydrolyze ATP more rapidly. It's a slightly different isoform of myosin, has faster ATP hydrolysis kinetics, and so it can go from cocked and flexed quicker. The other neat feature about fast twitch fibers is that they have the ability to remove calcium rapidly from the cytosol, more so than slow twitch fibers. Maybe they have more calcium ATPases, or they have other mechanisms in addition to the calcium ATPase, but they can remove calcium back into the SR much more rapidly than slow twitch. Fast twitch glycolytics are the true sprinter's fast twitch. These are what we also would use in maximal contraction, when we are absolutely going all out for full force contraction. They rely primarily on anaerobic glycolysis. Yes, they can operate oxidatively, and we can increase their oxidative capacity if we train a certain way. But their primary way of generating tension is through glycolysis and substrate level phosphorylation. These oxidative glycolytics, that's sort of your medium range. They're going to rely more on oxygen than the anaerobic fibers. They do rely on glycolytics to a degree. What we also see in these sort of medium phase, we see a little bit more reliance on creatine, that sort of thing. But I don't want to get into that too much. What we have here is sort of a true fast twitch and a medium, and the basis is the kinetics of the myosin, the kinetics of the calcium, and what type of energy source these muscle fibers primarily rely on. Now this is a big figure. This is one of the most important graphs of the entire semester. Before I get started into this graph, I'm going to tell you it's really important to be able to interpret this graph. What is this graph telling us? Okay. What is on the y-axis? It's the amount of force that a given muscle fiber, or muscle, shall we say, can achieve. And on the x-axis, the independent variable is sarcomere length at the onset of stimulus. Okay. In skeletal muscle, there are two major ways that we can modulate force of contraction. One is called recruitment. That is to say, how many motor units do we fire simultaneously? The more motor units, the stronger the contraction. The other major way that we can modulate strength of contraction is length of the sarcomere at the onset of stimulus. All right. So make sure that you understand this figure as it relates to what's going on in terms of, are the sarcomeres far apart, we have few cross bridges, or are the sarcomeres close together and we don't have any additional ability to shorten. Along those lines, these twitches here would be for sort of average sarcomere length. So if we have enough time to recover, we go back to 0. But that length tension relationship plays into this summation of twitches here. If we have one twitch and we recover, we go back, we get the same amount of force the second time, but as we begin to recover, we still have significant overlap in that sarcomere, and we get a stronger response on that second, more rapidly ensuing stimulus. That's the point of summation here. And if we have enough summation, we can get to maximal tension, what we call tetanus. And there are two major forms of tetanus. One is a more, dare I say, natural tetanus. That's because we're seeing a little bit of waggle here. Even though the muscles are achieving maximal tension, there's still a little bit of waggle as fibers cycle in and out. This is known as unfused tetanus. And you will still see an addition, a fall off of strength over time as fatigue ensues. A more artificial form of tetanus is called complete tetanus, where you have such rapid stimulation that the muscle fibers do not ever get a chance to cycle out, and that leads to rapid onset of fatigue. This is very hard to achieve in the environment. In natural situations. Examples might be certain types of neurotoxin poisoning, so forth. But where we normally see this complete tetanus, or fused tetanus is another term for that, is with artificial stimulation at a very, very rapid rate. And I did mention recruitment in a couple of slides earlier, so I just want to make sure you understand what recruitment is. Recruitment is how many motor units fire simultaneously in a given stimulus. And so what we have is the definition of a motor unit is one of these motor neurons and however many muscle fibers are innervated by that one neuron. We talk about large motor groups or large muscle groups. That's where you have one neuron activating many muscle fibers. When we're talking about fine movement, we're often talking about a very, very low ratio of motor neurons to muscle fibers, as low as sometimes 1 to 1 or 1 to a few, maybe a dozen. So large muscle groups often have large numbers of motor fibers that are, or muscle fibers that are activated by one neuron. And then small motor groups are where you have a low ratio. And this is why we can achieve fine facial expressions, fine movements in our fingers, and why it would be almost impossible if we could put a pencil lead on the tip of your knee that you could sign your name using the knee, because you have these large motor units which are not as good at fine movement. But recruitment is one of the major ways that we modulate strength of contraction. And we can also avoid fatigue if we're going at less than 100% by what's known as asynchronous recruitment. Let's say we have our grip strength and you're told grip at 75%, or let's say 50%. At 50%, approximately half of your motor units are firing simultaneously. And they will cycle out in order to recover and recoup energy sources, restore membrane potential, pump calcium out of the fiber. In the meantime, other motor units will cycle in and sustain that level of strength. That is known as asynchronous recruitment. Different motor units are taking turns maintaining tension, and it staves off fatigue. So again, the two major mechanisms of strength of contraction are recruitment, how many we recruit simultaneously. If we're not recruiting them all simultaneously, we can sustain longer because we have asynchronous recruitment. The other mechanism is the length of the sarcomere at the onset of stimulation. A little bit about mechanics of body movement. So we have different forms of muscular contraction, and yes, it's always generating tension, but there are additional ways that we can extrapolate this, and it does have implications on the response of the muscles and the development of muscles over time. For instance, we have what's known as isotonic contractions, and the definition of an isotonic contraction is that the load is moved. For discussion purposes, assume that we are moving the load at a constant speed. If we change the speed of that load movement, we are changing the acceleration of that load, and thus we would change the tone that we are firing those muscles. Okay? In order to accelerate something, we're going to have additional recruitment, and so that's going to cause a change in tone. For discussion purposes here, we're going to presume that we are not accelerating the load. So we're moving the load, and we have achieved a certain level of contraction tone in order to achieve that. And that's why it is called isotonic. Now, there are two forms of isotonic. We can have a concentric action, which is where we are gradually shortening the sarcomeres and the muscles. Or we have what's known as an eccentric action, where we start in a shortened phase and we gradually lengthen while sustaining our tension as we lengthen. It's called an eccentric or eccentric action. This is like a negative motion in an exercise, like for instance lowering yourself down from a pull-up bar slowly, or lowering yourself down to the floor in a push-up, or lowering a barbell down if you're doing a bicep curl. And they do result in different types of genetic and slow responses in terms of muscle readiness and development. Concentric, you can go for power, eccentric, a little more endurance. They do cause differential responses, and that's why you do recommend certain types of exercises depending on the type of goal that's going to be achieved by the person who's undergoing the exercise. So isotonic, we achieve a certain amount of tension that when we remove the load, we don't increase. If we increased, we would begin accelerating that load to a different speed, if you know what I mean. So there's isotonic, and now isometric. Isometric is where we are creating force, but we are not moving the load. So what's happening there is the sarcomeres continue to shorten, but what's happening is the force is being absorbed by these elastic elements inside the muscle fibers, including titin. Also includes the dystrophin proteins that we'll talk about with muscle pathology. But what they do is they absorb that tension. And so the overall muscle doesn't typically shorten, though you may see a thickening of the muscle as you contract. And you can do this just sitting here. Just flex your arm without allowing your elbow to change angle, and you will see that the muscle will get thicker. That's because the sarcomeres are getting shorter, but the overall length of the muscle is not. That's because the elastic elements inside the muscle are stretching to absorb that tension. So the sarcomeres shorten while the elastic elements stretch, resulting in little change in overall length. Now, these elastic elements do stretch during isotonic contractions. They are part of the muscle, Z-band to Z-band. So they're going to stretch there. It's just that more of that shortening is going to be translated into motion than what we see in isometric. So here's an isometric contraction. Again, the muscle is contracting, but the load does not move. We achieve maximal contraction, or whatever maximal force we achieve. Load doesn't move, and then we relax again. And so this is a nice little figure down here at the bottom. Now, the only thing I don't like about this figure is that it's showing elastic elements at either end rather than as part of the sarcomere, and I hesitate a little bit. We should be seeing those blue elements throughout the sarcomere because titin goes from Z disc to Z disc. But these other blue elements can include forms of dystrophin and other things that are also elastic elements. So here is an isometric contraction. So here's a muscle at rest, and what we see is that the sarcomeres shorten and those elastic elements stretch. The only difference is that with isotonic, the sarcomeres shorten and those elastic, the overall muscle shortens. With the isometric, the overall muscle does not shorten, only so in the isotonic. But you can see those serial elements are still stretching. It's just that we still have muscle shortening. So here, this is why you can bulge up your muscles the way you can. He's in a partially flexed situation there, so he's giving a little bit of shortening, but to exaggerate it a little bit. But if you held your arm straight out and you contract those muscles, you can feel the radius or the diameter of your muscles enlarge as those sarcomeres are overlapping a little bit. Some muscle disorders, just a few examples here. We're really just not going to break these out, but I am just going to tie some basic mechanisms that we've already talked about in here. For instance, muscle cramps are often due to either inappropriate calcium, residual calcium, or issues with the membrane. You're getting some sustained painful contraction because you've lost the ability to handle calcium correctly. There are issues with the calcium handling. Potassium is often common in this, and this is why they recommend things like Gatorade or some electrolytes in order to help sustain the appropriate membrane potential as well as calcium kinetics inside the muscle fibers. Overuse, what happens with overuse oftentimes is that you end up rupturing muscle fibers. That can lead to release of inflammatory cytokines and a sort of a painful stiffness, if you will, due to the fact that you have ruptured muscle fibers and you have to regenerate those muscle fibers using satellite cells. So overuse can lead to destruction of tissue that then leads to pain and inflammation. On the other side, given that we have evolved in a caloric deficit, muscles are actually somewhat expensive metabolically to maintain. So there is no ecologic benefit to having large muscles unless they are serving some sort of survival or reproductive benefit. You have to invest a lot of calories in order to sustain large, powerful muscles. And so we have developed a way of minimizing caloric expenditure if those muscles do not in fact serve a major purpose, and that is known as atrophy. That is, if you don't flex those and continue to use those muscles, they will stop expressing genes to help maintain readiness in that muscle, and they will also turn those molecules into fuel. There's no point in maintaining a large muscle mass if you're not using it. And so disuse will lead to atrophy. It's a normal mechanism for maintaining muscles where you need to maintain them in a caloric deficit, if you will. Now, if somebody is immobilized for whatever reason, then you may see pathologic forms of atrophy. There are acquired disorders for muscles. These things include things like muscular dystrophy, where you have issues with the dystrophin protein and it's no longer elastic enough. And so under normal exertional activity, you see widespread disruption of muscle fibers. So it mimics the overuse syndrome, only it's occurring at inappropriate times. It would be an example of exercise intolerance due to the fact that the muscle fibers do not have the elasticity that they need in order to remain intact. This second one is really, really interesting, and it's really interesting for a personal reason for me, because one of the former UND students actually has worked on McArdle's disease. He got his PhD at UND, and he helped uncover the enzyme, this myophosphorylase. He helped uncover that led to an understanding of McArdle's disease. What's happening in McArdle's disease is that you can make glycogen, but you can't convert glycogen back into glucose. And this is another form of exercise intolerance. Now, in this case, you don't have as much glucose available. You're depending more on blood glucose, and if your blood glucose is low, you're just not going to be able to generate energy in those muscle fibers. You're going to fatigue rapidly because of glucose deprivation. And so these folks have a fairly profound exercise intolerance because they can't convert that glycogen back. The other side of it is that their cells begin filling with glycogen, and that can lead to cellular disorders. So really, really interesting story. And that's the story here. This is Vinny Tagliabracci. He was a student at UND, graduated from the University of Indianapolis several years ago, and he thought he would go to medical school. Well, he interviewed. He absolutely bombed the interview. I've talked with him about it several times, and he just I think he knew in his heart he wasn't really all that interested in medical school. But he just absolutely, it wasn't because he was nervous. It was just he was flippant. I don't think he valued what it is to be a physician like many people would. So he didn't get in. He didn't have the best grades either. He was a little bored in undergraduate, so he didn't have the best grades. But he got into his graduate program, and he absolutely found his niche. He just blew up as a graduate student, discovered the glycogen issue, and then went on to a postdoc out in San Diego at a very, very prestigious lab out there, and then discovered a certain kinase that could have profound roles in gum disease. And so now he's got multiple millions of dollars in grant funding because he just is able to solve what are actually conceptually very simple problems that people have never solved. So I include this slide because I think it's always important to think about what would give you joy or satisfaction in your life outside of those relatively canonical, medicine or nursing or PT or OT. Think about these other things. We don't see these things nearly as often, but they can be as rewarding and lucrative as other careers. Okay, next phase of the lecture. We're about halfway through. we're going to talk about how we get from action potentials in the motor neurons to onset of contraction, the phenomenon known as excitation-contraction coupling. I'm doing this here, why? Because it's included as part of Chapter 12, but this also has implications in myocardial dysfunction. One of the scariest things I ever experienced as an EMT in an ambulance was what was called electromotility dissociation. We were seeing electrocardiograms on the screen, but there was no pulse. And so we're going to work through this mechanism. It's a multi-step mechanism. It's a big mech. We're going to work through it stepwise. So let's start at the very, very beginning. We have a motor neuron here that is synapsing with our muscle fiber. That motor neuron has an action potential. It comes down. You get those voltage-gated calcium channels that cause release of acetylcholine. So we have exocytosis of acetylcholine into the synapse. We have nicotinic cholinergic receptors. These are the nicotinic flavor of acetylcholine receptors, and they in turn trigger an action potential on the surface of the muscle. That action potential spreads down through all of those T tubules, and we get down deep into the cell. So you have this widespread cellular depolarization occurring from acetylcholine striking the surface in those nicotinic receptors. So here we have that action potential coming down, and as we get down into those T tubules, we encounter a really, really interesting little protein called DHP receptor, dihydropyridine, but you can just call it DHP. That's fine with me. DHP receptor is a voltage-sensitive protein. It is not associated with, it does not have a channel with it. It is on the surface of the T tubule, and it changes configuration when the voltage changes due to an action potential. Now, there is some research out there, there's DHP receptors also channels, it is possible, but in this place we're going to say no. Now, notice this little curly Q line right here going over here to this thing here in box 4. This is a physical, let's call it a quaternary interaction, so protein-protein interaction. What happens is when a DHP receptor changes configuration, it pulls on what's known as the ryanodine receptor, R, capital R, lowercase y, capital R. So here's ryanodine receptor. The ryanodine receptor is a calcium channel. So when DHP receptor pulls on the RYR, it causes calcium to be released out into the cytosol. And so once calcium is released into the cytosol, it binds troponin. Troponin moves tropomyosin, and we get power stroke on those myosin heads, so long as calcium remains in the cytosol. So again, action potential, don't leave depolarization up here. We had action potential, calcium leads to acetylcholine, action potential, DHP pulls on the ryanodine receptor, and we have calcium release. That leads to power stroke. Okay. Now we want to terminate this as rapidly as possible, just like neurons. When we have acetylcholine esterase and diffusion and reuptake, we need to get rid of that calcium very, very quickly. Otherwise, we would have sustained contractions that may not be useful in fine motor movements, like for instance, blinking your eyes, or fine motor movements in even your large group muscles. We want to terminate contraction as rapidly as possible, unless we continue to signal. If we continue to signal, let's keep releasing calcium out there. But if we are done signaling, we want to get rid of that calcium post-haste. That calcium needs to be gone. And so the main mechanism for calcium removal from the cytosol back into the SR is a calcium ATPase. It is a uniporter, one substrate calcium, and it is primary active transport. It is pumping that calcium back into kind of a form of reuptake, if you will, only it's inside the cell. We are pulling that calcium off. Calcium diffuses off of the troponin. Tropomyosin slides back into place, and we relax. It's not a very complicated mechanism. It just takes a few steps, work through it, and go from there. But again, one possibility for inappropriate or loss of contraction is a decoupling of excitation and contraction. So to summarize this verbally, we get a depolarization at the motor end plate. That's basically a synapse between a nerve and a muscle. That creates an end plate potential. Well, how's that created? Well, that action potential triggers exocytosis of acetylcholine. Acetylcholine binds to the nicotinic cholinergic receptors on the muscle, and that causes depolarization, an end plate potential. We get that depolarization, DHP, RYR, a calcium comes out, and as long as that signal up here is transient, we will get a single twitch, a single contraction relaxation cycle. But all of this going on, and partly why I've explained all of this, is that there are theoretical limits to how fast somebody can respond to a given signal. From an action potential coming from the brain, exocytosis of neurotransmitter, diffusion of neurotransmitter, binding the receptor triggering an action potential on the surface of the plasma membrane, going into the T-tubules, DHP, RYR, calcium diffusing out of the sarcoplasmic reticulum, getting to troponin, and then contraction. If you can do that, you got it made. Just what I did there, just sort of just walking through the steps. Well, all of that takes a couple of, few thousandths of a second, and that's called a latent period. And that gives you a theoretical limit to how fast someone could potentially respond to a stimulus. We talked about neural velocity in the past as part of that, and the number of synapses that can trigger fast or slow responses. Fewer synapses is faster. But we also have this latent period, the time required to get that calcium out and binding to troponin from that action potential is a latent period. And here is latent period manifested. So we have a probe here at the presynaptic terminal. We've got a probe in the membrane of the muscle, and we happen to have a tension unit for the muscle fiber. And so all three of these figures here are on the same time axis. So 0 is all here, and as time ensues, what do we see? We see a neural depolarization. That's going to trigger our calcium and neurotransmitter release. Okay. If we scroll down here, you can see that that occurs just slightly ahead of muscle fiber depolarization. So here's neural depolarization. Here is muscle fiber depolarization, and then we have calcium diffusing and binding troponin, and then we get contraction about within 1/100th to a 1/10 of a second later. So again, these are all on the same x-axis. That's latent period. The time from action potential depolarization in the nerve to actual contraction in the muscle fiber. And there is a theoretical limit there, usually somewhere around 2 to 5 milliseconds. Last section here, 7 more slides. Last section, we're just going to talk a little bit about smooth muscles, how they're classified, basic mechanisms, how they contract in a relatively synchronous manner and so forth, and then the way that muscle fibers generate force in smooth muscle. Now, smooth muscle is by far has a much greater variety in terms of contraction pattern, location, number, and so forth. So we want to talk about the various ways that we can classify these things. Different smooth muscles behave different ways in different parts of the body. So how do we classify smooth muscle? Well, we can do it anatomically by location. Vascular smooth muscle or gastrointestinal smooth muscle, urinary, respiratory, reproductive, ocular, your erector pili muscles, those types of things are all examples of location and lots of different locations. We will also classify smooth muscle by contraction pattern. We have what are called phasic smooth muscles, which are generally relaxed and then transiently contract. Skeletal muscles have a phasic form of contraction pattern, generally speaking. Okay, they stay relaxed most of the time and then they contract transiently. Then we have these really, really interesting smooth muscles that are called tonic smooth muscles that stay contracted, and they can stay contracted for hours and days. Skeletal muscle can stay contracted for seconds, maybe up to about a minute or two, depending on how fit those muscles are. But they got nothing on tonic smooth muscles. Tonic smooth muscles stay contracted for hours, days, even up to a week, and then transiently relax. The third schema for classifying smooth muscle is how the smooth muscles communicate with their neighboring muscle fibers. This is a bit of a molecular method of classification, but many smooth muscles, when we talk about the macro smooth muscles, have to contract as a single unit. This is also true for other muscle fibers, myocardial and skeletal. But we can solve it a couple of different ways in smooth muscle. And again, it's a form of increasing the diversity of smooth muscle. So we have different locations, we have different contraction patterns, and we have different ways of communicating. These all contribute to the vast diversity of smooth muscle. So I'm going to use examples of each of these things. I'm not going to get into this one so much. I'm going to get into contraction pattern and communication here in the next few slides. As I mentioned, phasic smooth muscles are usually relaxed and they will transiently contract. And the two major examples that we have of phasic smooth muscle are, for instance, esophagus, normally relaxed, and then we swallow and then we get a wave of peristaltic contraction and then relaxation again. Another example is the peristaltic motion in intestines and moving material throughout the digestive tract. One of my favorite words in all of physiology and medicine is borborygmi. I like sphygmomanometer, but I also like borborygmi. If you want to hear peristalsis in action, just lay on top of somebody's stomach with your ear on their abdominal wall. This is great to do with kids. Kids love this. You'll hear this little gurgling and bubbling. That's the smooth muscle contracting and relaxing in peristalsis. Particularly good after you've had a meal or something when peristalsis is very, very active. But you can hear little gurgling and bubbling all the time. It doesn't matter, and kids will get a kick out of this. Well, intestinal muscle is transiently contracting and relaxing, both in a radial pattern as well as a longitudinal one. Tonic smooth muscle is the opposite. It is usually contracted and then it transiently relaxes. So the contraction force is high, it relaxes for a short period of time, and then it resumes contracting again. The best examples of these are the sphincter valves in various parts of the GI tract. The sphincters remain closed most of the time, and then they transiently open to allow material to pass. And this can be for hours at a time at a minimum, sometimes days, even up to a week in certain situations where someone is withholding bowel movements and so forth. It's possible to sustain that. So we're going to look at the contraction mechanism of smooth muscle in order to see how we can explain that. One more example of tonic smooth muscle are your blood vessels. They are relatively, by definition, relatively high force, and then they transiently relax during systolic contraction of the heart. There are some really, really interesting exceptions to that in your coronary arteries and in your kidneys. They contract during systole in order to prevent that systolic wave from hitting the capillaries downstream of them. But in most vessels, what happens is you relax in order to absorb that systolic force, and then you contract again in order to sustain the pressure. Our third schema for classifying smooth muscle is how the smooth muscles communicate with other smooth muscle fibers. There are single unit smooth muscle cells and there are multi-unit smooth muscle cells. In single unit smooth muscle cell, what's going on is that these cells are interacting with each other through gap junctions, very similar, if not identical, to what's going on in the myocardium. So if we get some signal, we get calcium inflow, and I'll talk about contraction later on. Calcium inflow into this cytosol that can then spread through to calcium to cytosol of other cells, and you get uniform contraction, as if this was one big cell. How skeletal muscles they solve this problem is they fuse together and share cytosol directly. Myocardial muscles and smooth muscles solve this problem using gap junctions. The other way that smooth muscles can solve that sort of uniform contraction problem is by using neural structures. This is a neuroanatomical structure. I didn't mention this in the previous slide, and I'm not going to go back and re-record it. But the nerve endings in smooth muscle have a much more diverse structure. They have what are called varicosities. Instead of having this sort of podocyte or foot-shaped process, they have these little bulges where they exocytose acetylcholine. So in this case, the muscle fibers are not connected, and how we solve this uniform contraction issue is through this very complex varicosity mechanism of the neuron. So this is an example of multi-unit. The ciliary muscle in your eye is an example of a multi-unit smooth muscle. But again, we're talking about classification schema here. We have location, we have contraction pattern, and we have how the cells interact with other muscle fibers. Smooth muscle, in terms of its diversity, not only means location, contraction pattern, and so forth, but smooth muscle often has to operate over a wide range of lengths relative to, say, skeletal muscle or myocardial muscle. The reason that smooth muscle is smooth is that these sarcomeres, these contraction apparatus in the smooth muscle are not aligned in regimental form like the skeletal muscles or the myocardial muscles. They may run in various different directions, and you may get longitudinal shortening as well as lateral shortening in skeletal muscles, so they can run in several directions. It's a much, much slower contraction, and I'll show you that mechanism in a second. It's slower and it uses less energy, which makes perfect physiologic sense if you're talking about tonic smooth muscle that has to sustain contraction over hours and days, even up to a week. So using less energy, slower kinetics allows for contractions to occur over extended periods. Even in tonic or phasic situations, it's still a slower, longer process. So here's a comparison of the contraction timing. With a skeletal muscle, you can see it's very, very rapid, less than a second. Cardiac muscle, you might begin to approach 2/3 of a second in contraction. It's a little bit slower, and we'll give you that why later on in chapter 14. But look at smooth muscle, very, very slow onset. We might not reach full tension before 2 seconds at a minimum. And then relaxation out to 5 seconds or more. It's a very, very slow, low energy contraction relaxation pattern. And why is that? Well, there's a different mechanism for activating myosin, and let's talk about that. So here is the basic mechanism of smooth muscle contraction. The first event that has to occur is that we have a rise in cytosolic calcium. That can occur either from the sarcoplasmic reticulum. You can activate it through action potentials and RYR and that sort of a thing and get calcium release through kind of like what we do with the skeletal muscle. Or we can receive calcium from the extracellular fluid through receptor channels. This is why smooth muscle, and to a certain degree cardiac muscle, responds directly to hormone stimulation. Skeletal muscle does not respond directly to hormone stimulation. Have you ever been told something and the next thing you know your stomach goes into knots? Well, that's a hormone activating skeletal muscle contraction in your autonomic nervous system. That's why hormones can affect smooth muscle contraction through hormone signaling as opposed to through depolarization. We can also receive calcium from gap junctions. So gap junctions can cause a cytosolic calcium rise. So we got this calcium in here now. We've got the calcium there. It binds to a calcium sensor. You notice we do not have troponin anywhere in here. What we have, and if you've had cell biology, I'm sure you've heard of this protein. It's called calmodulin. It is a very, very common calcium sensing protein inside cells. Calmodulin, in the case of smooth muscles, will bind to calcium, and that makes it a cofactor for this MLCK, which stands for myosin light chain kinase. A kinase is a protein that will phosphorylate a target. In this case, it is the myosin light chain. And the myosin light chain is basically the hinge in the head. The heavy chain of myosin is the tail. I don't want you to worry all about that definition. I want you to think of it as a myosin kinase. So calmodulin and calcium serve as an activating cofactor for myosin kinase. And so when calcium and calmodulin are here, we phosphorylate myosin, and now myosin is in its active state. So MLCK is causing myosin to be phosphorylated. It is phosphorylating myosin. When it's in this myosin state, it will go through power stroke. That's part of the reason why it takes a couple of seconds for us to get to full tension. Now, there's some other reasons why we get into full tension here, is that this myosin contracts at much slower kinetics than the skeletal muscle myosin. It's a different kind of myosin. Different muscles, like for instance, if you have pork shoulder, if you have bacon, or if you have Canadian bacon, they all taste a little bit different. They're all forms of pork. If you have ham, tastes a little different. That's because you have different isoforms of myosin inside those muscle fibers. Okay? In any case, this myosin will flex and cock. Now, it doesn't need to have troponin and tropomyosin. Only the phosphorylation allows it to bind and cock and flex. But it's at much slower kinetics, a lower energy system. Also, don't worry about cyclic AMP. Yeah, it's involved in here, but I'm not going to worry about that. We just want the basic mechanism. Calcium rises, binds calmodulin. The calcium-calmodulin complex is a cofactor for this kinase that phosphorylates myosin and turns it on. Now, if we're going to turn the myosin off, so we have the ensuing contraction here, we have to have a phosphatase in order to dephosphorylate that myosin. So, finishing up here, just the last couple of slides. The things that we can do with our muscles are absolutely extraordinary in terms of how we lever them across joints, the oppositional pairs, the motor units, the kinetics. I love this picture because this is the picture that broke my heart. This was Cromartie intercepting Peyton Manning. This was the last game that Peyton Manning ever played as an Indianapolis Colt. But look at what he's achieving with those skeletal muscles. Never mind his ciliary muscles in his eyes and other things. It's absolutely extraordinary. The range of motions, the things we can do, and we certainly have skeletal muscle here in this pose, but think about digestion and exocrine function. It's absolutely extraordinary what we can achieve physiologically with these muscles, the way they're configured, the way they contract, and the way they behave. And I think that's the really cool thing, is when you begin to understand how muscles really work, you can begin to see the extraordinary range of possibility in human beings. And so that's it for this chapter. Again, thank you for your time. If you have any questions about smooth muscle or excitation contraction, let me know, and I'll see you when I'm looking at you. Bye.